but we have a short today guided model building exercise. So we talked a little bit about this at the beginning of the day. The way that the fourth module is going to work each day this week is we're going to have prepared an exercise for you to do that will be a little bit more self-paced. So we usually have 10 to 15 minutes of setup. I'm going to go through it very quickly today so you have plenty of time to work. But we'll have 10 to 15 minutes of setup of a biological problem that has been modeled in CompuCell, followed by an exercise with a number of questions that you can do. Like I said, kind of at your own pace, and the instructors from each day will be in the room to kind of move around and help people as they may need it. That reminds me, James, we may need to make a couple of breakout rooms for people to join. We probably won't need them today, but for future meetings, we will use them. And we can always make one today if we need to for individual questions. So like I was saying earlier, if you have any questions about the exercise you're currently working on, or questions about the day's content, anything is open to be discussed. We may check in periodically specifically about the exercise to see how people are doing. But if you have any questions about anything, the fourth module is a time where you can get that addressed, including troubleshooting and problems and things like that. So just as a quick reminder, this session, like all the other sessions, is being recorded and live streamed. And we will go ahead and get started here. So just some learning objectives for today's activity. I do want to quickly share links. So these slides have gone up since lunch. I had to upload a new version. So if you have not downloaded these yet, here is a direct link to the slide. And then here is a link to the code folder, like for Juliano's exercises, or excuse me, Julio's exercises. Can you tell it's the end of the day? I've forgotten how to speak. So learning objectives today, we're going to talk about a cell sorting example that you can walk through some questions with on your own. We're going to cover the very basics of cell sorting modeled in CompuCell. But the main point today, and some of this is going to be a little bit redundant after Julio's session, but this is mainly to get some more hands-on experience working with a CompuCell simulation, specifically working with modifying the CompuCell code. The other goal is to become familiar with the variety of the CompuCell code plugins, like Julio was doing in the previous session, how they control and interact with the simulation, as well as to explore how various parameters affect simulation performance and behavior. So the point here is more to get familiar with interacting with the CompuCell interface more than anything than it is to understand the biology of this particular problem. We're going to be working a little bit with the contact energy plugin in this exercise, but like Julio said, we're going to talk in more detail about the actual biological basis of contact energy tomorrow. Today, it'll be mostly in the abstract. So the simulation we're looking at today is an aggregate of cells, 2 cell types that start randomly mixed, and because of differential contact energy, end up sorting themselves into groups of like cells. And in this case, you can see one cell type by the end of the simulation has kind of engulfed the other from an initially random startup. This is a phenomenon that happens in nature, particularly during embryonic cell development. Sorry, that pop-up was groups or breakout rooms. And this engulfment behavior does typically happen in stages for various different organs during development. So we're looking at a very simplified version of that today. One way of modeling this, so there's some disagreement in the literature about what's the most useful way to model cell sorting behaviors. The way that we're going to be looking at today is the differential adhesion hypothesis, which states a couple of kind of base assumptions, that there's a hierarchy of adhesion energies when cells come into contact with other cells and with their own cells. This adhesivity is based on the number and type of cell adhesion molecules in the cell's membrane. But for our purposes today, this can be abstracted basically into a scalar interface value where each interaction type between cells has a specific contact energy, like you were using briefly in the previous session. States that cells fluctuate randomly within the aggregate, that they don't grow or change their properties during the sorting. And that, like I talked about before, the less adhesive types tend to engulf the more adhesive types. I'm actually going to skip over this for now since we're running out of time. But like I said, these slides are available, and please do go download those now, because the slides act as kind of the handout for the activity for the next half hour. James makes a good point here that breakout rooms are meant to be used to travel basically with an instructor or with a group of other students to discuss. We can't communicate easily with you once you've moved into a breakout room, and we are also not directly notified when you move into a breakout room. So if you're moving to a breakout room hoping for help without asking somebody first, we may not even know that you moved in there until we actually look at the breakout room roster. So that's an important note. And like James says, we will review these ideas about cell sorting in more detail in future portions of the workshop. But I don't want to burn too much time on this here. Basically, the key takeaway is that the algorithm tries to minimize total system energy by reducing the overall contact energy within the energy equation, and that systems of 2 cells within a medium have 5 contact energy values. We'll see in a minute that the simulation actually has 6, but there are 5 in the mathematical model here. 1 energy between cell A and cell A, 1 energy between cell A and cell B, 1 between A and medium, between B and B, and between B and medium. So basically each permutation of, or not skip permutation, each combination of possible cell interactions has a contact energy value. And that the system will try to arrange into a pattern that minimizes the total amount of contact energy within the system, within certain constraints, obviously. So just a quick video of what the simulation will sort of look like once we get it built. And as you can see, they start in a random configuration and slowly engulf one another. Obviously, in the real world stochastic situation, it's not a perfect engulfment. There are these groups that kind of hang out in the middle until they, find their friends on the outside and disperse. We do actually have, there is a tool on NanoHub that will just run this simulation within CompuCell. The version of CompuCell it's using is kind of outdated at this point, but if you did just ever want to look at this and mess around with it without having to rebuild the simulation from scratch, that link is in the slide decks here. So what I think I'm going to do is, I was originally going to build this with you, but since you just got a bunch of experience working with Wizard with Julio, and we have instructions here, I think what I would like to do is actually just go ahead and turn this over to the room. So like I said, please do go download those slides. I will share the link again. And just to give you a quick overview of what this is going to look like. So there's some text instructions here about how to build the simulation. It's very simple. Again, if you have trouble, please contact one of the instructors in the room. We can walk you through it. You're basically just building a simulation with 2 cell types, and you're choosing the contact plugin and the volume flex plugin at the start. And that's it for the setup. The demo code that is provided is the complete model build before the exercise starts. So if you would like to just open that instead of having to go through the Wizard process, you can do that as well. So Sharon has mentioned that the slide link that I'm giving takes you straight to the slides. If you go to Save As and download as PowerPoint, that's a good point. So if you are having trouble with that, you can get to it from the normal student materials folder, which I'll put in here again, just for ease of access for people. And it'll be in the module slides folder. This is module 1.4. You'll also notice that in the demo folder, along with the demo code, there is an additional PDF with some additional information about the differential adhesion hypothesis that describes the mathematical model in a little bit more detail if you need to look at that at any point for reference during the course of the exercise. But basically just take a couple of minutes here to get the simulation built. I've already had experience with this from Julio's lecture. This will point at a couple of important plugins, and then it'll have some questions for you to start answering. So just go ahead and take the next, so we've got 25 minutes before 6 o'clock. Just work through as much of this as you can or as you want. So this isn't necessarily something that you need to do linearly. If you get to something that you're not, there's a number of different questions to answer here. So if you get to something that you're not super sure how to address, feel free to skip ahead. Again, this is kind of self-paced. So just however you feel you can get the most out of this exercise. Feel free to jump around the slides and ask questions about any of them. You don't have to do this in order. I'm actually going to go ahead and stop the share here so I can see people's screens. But the floor is now open for questions, again, about this exercise or about anything that's been discussed today. Yeah, like James says, there's a number of different things you can do here. There's some suggestions with some hints about how to do the things that are suggested in the slides already. So again, if you get stuck on one thing, feel free to just move on to the next thing. There's plenty of different options to try.
And you should probably leave the slide up though, because people don't have the instructions.
Yeah, I'll go ahead and I'll share the model building slide, because that's probably what people need right now. Let me reshare this presentation.
As I was saying, there are a lot of things you could do. So I strongly encourage everybody to get this model working and then explore what happens when you change the contact energies. Because there are many, even with those two cell types and the limited number of contact energies you have, you can get at least 6 or 7 different biologically meaningful configurations. And I've put in the chat a couple of possibilities. I'm sure Hayden also has them. But once you have the simulation running.
So Pete asks, the code in the Google Drive, that is just the starter code. There is no technical finished exercise here because there's so many different questions people can answer. If people are interested in getting final code for any of the questions here, we can certainly generate that. It may take us a day or two to get that organized, but we'd be happy to help walk people through the end of an exercise. Wilson May says the CompuCell3D version is too old for this. Were you trying to build the exercise yourself or open the provided code?
That's not an error to worry about. It's a warning that simply says that the code you're using is old. But as long as it runs, it should be all right.
Yeah, so I actually, CompuCell now has an auto updater feature that I ran last night. So I may have a new hotfix that you don't, and it may just be a very slight version mismatch. So if it's running and everything seems okay, I wouldn't worry about those warnings. Obviously, if it doesn't run, please let us know, but go ahead and move on. I do want to address a couple of these questions as a group, maybe to get a little bit of discussion going. So when you run the simulation as built, what happens? Melanie has a good question. Why does setting neighbor order as 2 and setting up the model still show neighbor order as 4 in the XML? It's good. I don't know if James has a clean answer for that. We can circle back around to that, Melanie. All right, so we've got doesn't sort cells, the cells do not aggregate. Yeah, so that's basically, yeah, I'm not sure why that's happening, Melanie, with it showing neighbor order as 4 in the XML. That may be a bug that I've not encountered. Maybe somebody else can elucidate more in the chat, but yeah, so we've got some people saying it doesn't aggregate. So does this make sense given the default volume and contact energies and based on the energy minimization? Somebody want to elaborate on that a little bit? Basically, why don't they do anything? There's been a suggestion that we build it live. We can do that. Let me remember how to use Zoom. There we go. We're going to close this. And so when you launch CompuCell, or if you're in NanoHub, you'll have your player window. So we've got their response, the energy. It does make sense. The energy is the same for all combinations. So the total energy is already at a minimum. Yes, exactly. So you launch CompuCell. I'm just going to rebuild this live, like you're saying. The screen share working. You see the split screen. I've already got Twedit open, but normally you would click here to open Twedit. In Twedit, you will go to CC3D project. We're going to start a new CC3D project. I'm going to call it cell sort 08. Again, try to avoid using dots in your file names. I'm going to put a 2 on the end of mine because I've already done this a couple times today. We'll leave Python plus XML checked. Click next. Here you can modify all of these values. The only thing that we're going to change is pixel copy range, neighbor order of 2. You can put 4. Basically what this does is it just affects how quickly the sorting happens because cells can see a little bit further away with each Monte Carlo step. We're going to use 2 for now because that's what's in the slide instructions, just to make sure we all end up with the same thing. And then in the initial cell layout, you'll start at rectangular slab, but you want to change this to the blob initializer. And this will set the cells up in a blob in the middle of the simulation. So we'll go ahead and click next on that. Now we need to add our 2 cells. This is pretty straightforward. I'm gonna call them cell A and cell B. So you'll add cell A and cell B, and you can also just hit enter to add your cell types. Make sure you do hit enter though, because if you don't, it will not put it in the list. Next, we are not using a chemical field in this example, so we can hit next. And then in our cell properties and behaviors, we'll choose contact and volume flex because we want contact energy and volume constraint plugins. Yeah, Pedro is reminding us no numbers in the beginning of the file name. So no numbers, no periods, no hyphens. Try to stick to all letters, numbers not at the beginning, and underscores. So again, contact and volume flex here, because we can add plugins later through the menus as we need to. And that's it. That's all we need to get our simulation started. So I will double click on this to get it open here. And then we want to click on the XML to look at our specifications file. And I'm going to go ahead and open this in Player just so we see how it runs as is. And then as people were saying, this is the behavior that everybody else got, right? Where everything kind of sits in place. And again, like we said, that's because all of our contact energies are the same. They all start at a default of 10. So the whole point of the differential contact energy is in the name, right? So the next step would be to initiate the sorting. So if people want to take a minute and think about how to do that. You know, I've already hit on it a little bit, but in the interest of time, it would just be the reverse of having all the values be the same, right? We need some of these values to be different so that we have differential adhesivity. Yeah, so Julia makes a good point. Contact energy low between equal cells. If we look at our initial sorting plugin, how do we want these values to be on a relative basis? We don't need exact values for these. But which values do we want to be high and which values do we want to be low? Again, to attain the behavior of like cells grouping together within the aggregate. Okay, so that's a good start. Medium would be 0. Basically no energy state. Yes, so Pete makes the point, choose one cell type to have a high contact energy with the medium. So Pete, this would encourage what kind of behavior? If only one cell has high energy with the medium. And you're welcome to unmute. I think everybody should be able to unmute at this point. If not, we can help you unmute. But if you prefer just to answer verbally, you can also do that.
At this point, it's been a while since I ran my simulation, but I think it was either fragmentation or migration towards the edge.
Mm-hmm. Right, so if one cell type has a high contact energy value with the medium, it's going to, on average, try to avoid the medium because the system is looking to minimize its energy, right? And that's where we end up with the engulfing behavior that we saw at the beginning, because one cell type tries to avoid the medium and the other doesn't mind being against the medium so much. So to give some initial sorting values, so Pete's basically on the right track here. Let's see, we have higher values between different cell types, lower values between the same cell type, and maybe default with medium. Yeah, so these are some starting values to try. So if you want to look at your contact plugin, and again, I mentioned here we have an additional value that's not represented in the mathematical system, and that's the medium case. Because that's 0, you wouldn't normally need to represent that, but CompuCell still needs to know what the medium contact energy is for parameterization purposes. because medium is treated as a cell type in the lattice. So that's where the extra, the sixth value comes from in the computational model that is not necessary in the mathematical model. So once you've got these values changed, got about 8 minutes here. Go ahead and move on to running the simulation again. Actually, let me just do this on my end. I'll go ahead and change these values. And there's a number of different ways you can parameterize the contact energy to result in the sorting behavior, some of them even faster than this. This is just a good starting point. I think these might be based on some literature from the original model. And now if we stop our simulation, if you're running on NanoHub, you can pause and restart. And then you every time you make a change, like with what Julia was doing, you have to do the save all. Make sure all your files are saved before running again. And then here we do start to see the sorting behavior. You can see the cells more or less immediately find like cells and join up into groups. Now this version of the simulation runs a little bit slowly, so you do start to see engulfment behavior, but it takes a very long time to get there. There are a couple of different ways you can speed that up. Anybody might want to put some suggestions for that in the chat. He says increasing the step size / intervals. That would speed up the rate of animation. I guess it would work. So Gulsemi, you may have forgotten to switch to the blob initializer. If you look in your XML file, you should have blob initializer here. It may, if it says uniform initializer, then you may have forgotten to swap it. It may be the uniform initializer creates a square grid of cells with a random distribution. The blob initializer creates a circular blob. So while that runs, yeah, so here, what parameters can you change to speed up the sorting process? We'll just look at a couple real quick because we are running out of time here. I know people are getting tired today. We'll go ahead and stop. So one thing we can do is we can make one cell type very afraid of the medium. And you'll see that the green cells, which in this case is cell B, sort of immediately form a perimeter. One of the side effects of that is that it does tend to trap the blue cells, cell A, in these pockets that will eventually sort back out. Let's try again with it slightly less, say 20 here. Again, these values have to be floats, to keep that in mind. And let's make this one, we'll set this one back to the default of 10 and see what happens. Anybody have any thoughts on why this one did not create the intended effect? Yeah, Julia says afraid of is nice for understanding. So there's always a little bit of a threat when you try to anthropomorphize these things, because it is a statistical probability process. But one way of thinking of it is that when the contact energy is high, because the system is trying to minimize its contact energy, it will try to minimize the number of contact points between combinations that have that high energy. So the cells do move away from the medium as though they are afraid of it. But again, keep in mind that this is not a conscious process. So one of the reasons here for not having the engulfment process is the cell B against medium is actually lower than the cell A to cell B. So again, like James was saying, there's a lot of different ways that you can play around just with these contact energy parameters and get different simulation behaviors. And we have a couple of, sorry, somebody breaking in?
So Hayden, maybe go back to the player for a second.
Mhm.
And expand player a little bit, so it's easier to see. We can. Okay. Now on the top left where it says model editor, if you click where it says plug in contact. No, on the top left where it says model editor. Okay, that's good. Where it says model editor, click where it says plug in contact. Go down.
Yes.
Plug in
So you can
contact.
change these in real time.
Yeah. You don't have to restart it. The one thing you have to remember here is that if it's written as a float, you have to type a float. So if it says 20.0 and you want to make it 19, you have to type 19.0. So you could do this while the simulation's running. The thing is, it doesn't log it, so you have to remember what you typed. But for example, if you wanted to make the cell, the type 1, type 2 at contact energies low, and remember, don't hit stop, hit pause when you do this. So why don't you see what happens if you make the type 1, type 2 contact energy very small? And you may want to zoom in player so you could see that better. There you go. You notice they're trying to maximize the contact rather than minimize the contact between the two cell types.
Mhm. Yep, and you still have all cell B around the edge. So there's still, like, I guess, this minor engulfment behavior with maximized contact in the middle.
There are at least a dozen different behaviors you can get just with this system by changing these contact energies. All of them occur in biology. Every single one exists. Some of the differences between the patterns are pretty subtle. Oh, that's a very good question, Venceslas. We'll talk about it tomorrow, but think about what energy differences mean. Remember, absolute energies never matter. The only thing that matters is the difference in energy between, in this case, boundary and boundary. And so if you create boundary between cells and medium, you're decreasing the amount of energy between that, the amount of contact between that cell type and itself. And so whichever cell type has a higher contact energy with itself will tend to go to the outside. So now if you make cell medium contact energy larger again, Sorry, not cell medium, cell 1, cell 2 contact energy larger. The one that you edited before.
How much larger? Would you say?
I can't see your value. 16 is your value for cell.
It's 5 right now.
Okay, so make it 5.
Well, that's what you said, make it very large, right?
Well, so make it, but start out by 5 will be fine. Okay. You'll notice the green cells are now aggregating. There you go.
We just up it by 5s. Yeah, so as you increase the cell contact energy, the cells tend to group into
Yes, absolutely. There's a
structures of like
question.
cells.
Yeah, sorry. There's a question will depend on the contact energy between A and B, A and B and B. Absolutely. All of them matter. Again, if you divide all of them by a constant, it doesn't do anything. Or if you add a constant to it, it doesn't do anything. But the relative values matter. If AA is equal to BB, then the only thing that matters is AB relative to that. If AA is bigger than BB, then what matters is AB between AA and BB, or less than both, or bigger than both. And actually, when it's in the middle, it matters whether it's less than the average or more than the average. Now there you've got it, where you're going to disperse into the medium.
Yeah, here I just gave both cells the same affinity for the medium.
Yeah.
And the cell energy is very high.
So you could even go further, make the cell medium energies a little lower, and the cells will run off into the medium. So you just changing those 4 or 5 numbers, you can actually get an awful lot of different behaviors. Notice here's a good question. Pause that for a second, Hayden, and then let people think about this. Why is it that the blue cells are clustered inside of the green, but they're dispersing in the medium? Well, no, there's an energy with medium. You've defined an energy with the medium.
Actually, to build on top of that, I've also given cell A and cell B the same affinity, and you'll notice that when it is running, even though the blue cells that have the same affinity have disconnected, the green are still grouped together.
Well, that may be true, but there's something else going on, which is if I'm a blue cell inside the green, do I know that the medium is there? So let's wait. If the blue cells break through that green barrier and touch the medium, what's going to happen? You see the cluster of blue cells at the top inside the green? They're going to spew out. That's right. That may or may not happen because the green barrier is getting thicker. But if by chance the blue cells come into contact with the medium, they'll escape. As long as they're inside the green, they'll stay contained. That's like a bursting phenomenon where you have a cyst and the cyst bursts.
Peter suggests increasing the temperature a bit. This just effectively
Make-
speeds up the a little bit more.
Now, one thing that would happen is if you make the blue cells want to coalesce again, for example, you make blue medium contact energy bigger, will the cells immediately cluster? Yeah, that's what I was saying. It's a little bit small on your screen, Hayden, to see that.
Yeah. I don't know if there's a way to
If you
zoom
just
in on
read,
the-
if you just read off the values, that's probably okay.
Yeah, so we've got medium cell A, medium cell B, the same at 5. Cell A 16.
There you
Cell
go.
B is 20. Cell B is 20.
That was the bursting that Sharon wanted to see.
Mhm.
Yeah. Yeah. But now if you made blue medium contact energy larger, so they want to aggregate, see what happens. So really, it's a good idea for you to play with this on your own too, because there's a lot to explore. You'll notice the blue cells are aggregating. Why don't they cluster immediately? Anybody? Why don't they clump up all at once? Well, somebody says because the BB energy is still low. You see that they do cluster. Right, there we go. Contact happens randomly once they're apart. It takes time for them to find each other. That's right. If the T is too low, it takes them a long time to move. And they don't find each other. And so this is a random process. This is what's called an activation process. It's like the time it'll take that blue clump to come out of the green, to get its way out of the green. It'll do it eventually, but it takes a long, long time to break through a barrier. If you've done chemistry and you know that there are reactions that have barriers to the reaction happening, the barrier height gives an exponential time to the time for the reaction is exponential in the barrier height. This is something that happens a lot. When you're thinking about these problems, one of the things you should think about is how do cells know? How do they sense signals? What is the information that's actually available to a cell in the real environment? A lot of the things that we can do in an engineered world, like send signals over long distances or look, are not available. Cells really only know what their local environment is. They know their internal state and their local environment. And so even if they're two clusters of cells, if they're separated from each other and they're only interacting by contact, there's no way for them to know that there's a cluster somewhere else, even if it's quite close. Now, there are ways of cells sending long-range signals through the extracellular environment. They can send diffusible chemical signals. They can send electrical signals. They can pull, if the extracellular environment is a material, they can pull on it and send stress signals. But when you're building a model, you should always think about how signals, where signals come from, where they go, and how they're received. Because it's very easy to design simulations that replicate what you see in biology. It's much, much harder to design simulations that replicate what you see in biology that actually respect the limitations that exist in biological systems. And so that's one of the reasons that it's quite important to think carefully about what it is that you're trying to do and what the cells that you're talking about can do. And again, this simple example is a great way of exploring that. It's an awful lot of richness in this simple problem. How did you kill your blue cells? I missed that here.
I didn't. It's emergent behavior. I changed the neighbor order to 8. I was hoping they might grab each other quicker because we are technically out of time here, so I don't want to take too much longer.
Changing the neighbor order to 8 effectively increases the contact energy a lot. So it puts pressure on the cells. And if its pressure is too large and lambda volume is too small, they'll collapse and disappear.
So like we were saying, kind of a quick overview, and if anybody wants to spend some time working through some of the activities we have here, you're welcome to do so. And if you have any questions along the way, please do email us. We'll try to get back to you. So it's a good exercise of adding a third cell. See if you can find a configuration that creates concentric engulfment. You can rebuild the simulation in 3D. And then I had a little bit of a priming exercise for the rest of the week about if you wanted to track a single cell moving through an aggregate, starting to think about how you might do that by using some plotting tools. But we will get to some of these things starting tomorrow. Any questions before we, well, I guess technically we're in the Q&A session now. Let me go ahead and stop the share.
So we'll launch a module questionnaire, but people are free to stay and ask questions about this or anything else. You can screen share as well. So if there's something you want to show us or show the other people in the course, you're welcome to do that. Some result you're interested in, some problem you're having. Those are all possible. If you want to debug something technical about the software installation, I recommend going and seeing whether somebody can help you, like Gilberto or Julio, and then going into a breakout room for that, because that's pretty specialized. But any general questions, please feel free. You should be able to unmute, and you should be able to screen share if you'd like.
The Slack channel is also generally a good place to put help with debugging. If you're encountering an error, chances are you're not the only one, and someone else who is experiencing it may have already solved it. So it may be it's a little bit luck of the draw, but it may be one additional way to get help a little bit quicker than we can outside of hours during the workshop.
And maybe it's worth saying that this was the simulation that CompuCell was written to solve 30 years ago. This was the specific example that this technology was developed for. I don't know. When I was a student, I found the history of science rather uninteresting. As I get older, I find it more interesting, maybe because I have more history of my own. But sometimes it does help you understand why things have the structure that they do. Sometimes that's something useful, sometimes it's an annoyance, but very often we're prisoners of our history in one way or another. Did anyone come up with any energy configurations that they think are interesting or worth exploring or worth sharing with the others?
I think Pedro says that he can't unmute.
If you would like to unmute and you cannot unmute, please put that in the chat and we'll make you a co-host so that you can unmute.
Oh, sorry. He was telling me to mute. Somebody was getting feedback through the microphone. I guess it must have been me.
Can I share? Whoa. Maybe this one. Does that work better?
Yes.
Sorry.
No
To
longer
log in
on.
twice, you have to be careful about
Yep.
that.
Yep. Microphone on one machine and speaker on the other doesn't work. I just wanted to share an observation about this Steinberg hypothesis. Because when I was in graduate school, I was working with one of the premier scientists of morphogenesis, Ray Keller, who looked at everything having to do with morphogenesis in Xenopus. And in our early graduate developmental classes, and as well as in the lab, he used to kind of make fun of Steinberg and say, well, Steinberg thinks that all this happens passively, and he doesn't appreciate that it won't happen unless cells like do active things. And so we all had a big laugh about it. Then some years later, I built my own POTS model based on James's early papers, and I recreated the simulation. In the process, I went back and I read a couple of Steinberg's papers, and I was really pleasantly surprised that is actually quite genius and amazingly clear thinking. And he wasn't saying that morphogenesis happens passively. He was saying that if you don't understand what will happen without active behaviors by the cell, then you can't understand what happens with active behaviors by the cell. You have to understand what will happen passively on its own first as your sort of ground state. It's kind of eye-opening.
Yes, well, that tends to be the case. I mean, Walpert tends to be caricatured as the person who came up with positional information theory. Actually, if you read Walpert, he says that that's one element of many in developmental processes, and he never insisted on positional information in the way that people caricatured it. And Mel Steinberg was a great scientist, and he was very aware of the complexity of development. But he also had the insight of looking for a simple mechanism that could explain some things. And then I think this is a philosophy in these kinds of models. You come up with very simple hypotheses, and then you push them until they break. You don't start out with the most complicated set of hypotheses first. And Ray Keller is also somebody who was very much like that, coming up with a set of very simple mechanistic hypotheses that you could explore, and then trying to understand their limitations. So I think, of course, you work with a lot of the great developmental biologists and theorists. So you have a special perspective on that. Are there comments or suggestions? Anybody else want to share anything with us, or any questions that you'd like to ask? Julio, go ahead.
Just make a comment on top of Sharon's comment, and I'm not sure if you are aware, but on the topic of cell sorting and also others, there's some commentaries by Albert Harris from UNC Chapel Hill that are also very enlightening and very useful. He made very good and valid criticisms for the Steinberg hypothesis, very grounded. And he put forth so many other theories for the cell sorting that later only recently got up by other people, and they never cite him. So the idea of, for example, is given by surface tensions of the cells. It was something that was put forward by Albert Harris first, or the cell pushing and other criticisms. And I have seen people, a model from Canada put that forward as was his original idea. It's not. And now more recent from people okay, that's not single out those people, but from New England, right? That are also pushing these ideas as is their original ones, and then it's clearly not. He's already been discussed before by Albert Harris. I think he's still a emeritus professor at here, UNC Chapel Hill. I never met him, but I should talk to him someday. So if you don't know about his work and his commentaries, I suggest you take a look at it. It's also a good reading.
So there was a comment about tracking cells. There are a couple of different ways you could interpret the comment about tracking cells. One of them would be to measure the contact between cells or the correlation between cells or the cluster size. And we'll talk about that a little bit tomorrow. Another one would be actually to look at the trajectories of cells. Pedro will show you later in the week about how to track cell trajectories. You can create a dictionary or a list which keeps track of the position of the cells over time, and then draws the trajectory between the cells. It takes a little bit of coding to do that. It's not automatic inside of CompuCell, but it's possible to do that. I mean, another thing that's interesting is to ask the question, if you take the same simulation with different numbers of cells, how long does it take to sort? It does depend on the size of the simulation. If you change the fluctuation amplitude, the so-called temperature, how does that affect the sorting time? That's another interesting question. There's really quite a bit that you can look at. A lot of these things are much easier to study in a simulation than an experiment, for a variety of reasons. But the two speak to each other. The differential adhesion hypothesis came out of experimental work, but really understanding its meaning in terms of physics came out of modeling and simulation.
Julio, if you need Julio Hor, I was talking to. If you need to implement this type of tracking, cell tracking, you can ask us to help you in your simulation, and you can present it in the hackathon. So if you need it soon, we help you implementing, and you present as a hackathon project.
Any other questions? Our last 5 minutes here. All right, well, I think we'll wait here till 6:30 just to be on the safe side in case anybody's still typing anything out. But if there aren't any more questions, I think we can officially call that the end of day one. And we will see you back here tomorrow at 10 o'clock for day two, where we start to really dig deeper into more of the biological side of things. Sammy says, can you share your last simulation XML file again? Yes, can. Just the XML file, or do you want the whole simulation? Because the changes we were making at the last minute, they were all in the live player, and they won't be saved in the XML file I would actually send.
And I think one of the things that's important here is you're not going to break anything by playing. At worst, CompuCell will crash and you have to relaunch it. And so I really encourage you to play, to explore the values of the energies, try it out. Don't wait for us to do it in front of you. Try it for yourselves and see. Try it always. Ask the question, what do you expect when you change a value? Do that before you make the change. Think about it for a minute. Come up with a guess of what's going to happen or a hypothesis about what's going to happen. Then try it and see whether you were right. That's a great way to build up biological understanding. This is a tremendous way of exploring a biological mechanism very quickly and building your understanding of what biological systems can do and how they do it.
Yeah, so you'll say me, if you follow the instructions in the slides, you should end up with the same simulation. Like I said, I think that the problem was just forgetting to switch over to the blob initializer, but I'll make sure that the links to the slides and to the XML file or the simulation file go out in our evening announcements email tonight, which we'll start sending out this evening. So that you do have access to the properly initialized file. What is the name of what, Pedro?
In this particular case, whether you use blob initializer or uniform initializer doesn't matter. You're going to get the same results. It'll just take time for the initial square to turn into a circle.
That's my cat Madeline making a rare appearance, Pedro. Normally I have the door closed so she can't get in, but I accidentally closed her in today.
Yes, well, as I say, there are a lot of situations you can get. As I say, there are about a dozen different situations. One cell type disperses, the other clusters. Both cell types disperse. One cell type disperses in the other, but they stay in a cluster. One cell type goes to the center and the other goes to the outside. They form clumps, but they don't sort. All of those are possible. All right. Well, I want to thank everybody for sticking it out. It was a long day. I hope we'll see you tomorrow. We'll analyze this problem a little bit in terms of thinking about it in the morning, talk a little bit about the principles behind how you could think about organizing this, and then we'll get back to work building models. So I want to thank you again for coming.